Okay, cell biology fans, here is the next lecture in the list of things to do. We need to talk about endocytosis. I've split the PowerPoint into two so that I could talk about the main mechanisms of endocytosis prior to talking about the specific examples of endocytosis. And as you all know, endocytosis is the process of internalizing molecules from outside the cell into the cell. And there are many different ways that this can occur. The one that I'm going to talk about mostly is receptor-mediated endocytosis. That's using a receptor on the outer surface of a cell. So if I draw here an outer surface of a cell, you might have a receptor that's a transmembrane domain containing receptor that sits with a portion of the receptor in the extracellular space. It can bind to a ligand, so ligands bind to receptors. And when the ligand binds, it then causes the entire receptor to be internalized into the cell. That is receptor-mediated endocytosis. There are special cases of endocytosis called phagocytosis, and we've talked about this word phage. Phage is to eat, and this is cells eating cells, and this actually results from a cell that can send out cell membrane. So they basically send out cell membrane and encircle a particle. So in this picture, right, a blue particle can be encircled by membrane. That membrane can then fuse, and if the membrane fuses, up here, oops, sorry, that should be in red. If the membrane fuses, what you end up with is a vesicle that's inside the cell, and that is called a phagosome. Phagosome. It actually is very similar to an early endosome. All right, so that's another process whereby molecules can be from the outside in, and the last one that I'm going to mention is pinocytosis, and pino is cell drinking, and that means that fluid can be taken up from the extracellular media by membrane-bound vesicles. And these are very small vesicles, and in some cells this happens 24/7, 365, and it's a process whereby a lot of material can be sort of sampled from the extracellular media in a process that allows the cell to understand what's going on in its extracellular environment. The endocytic pathway is composed of the early endosome, the late endosome, and the lysosome. And I should tell you now that the late endosome often is called a multivesicular body. Multivesicular body. And you'll see why when we look at it. You can even see it here, right? It has lots of vesicles inside the actual late endosome itself. And this endocytic pathway can start either at the plasma membrane, as shown here. So you have the plasma membrane and vesicles budding in. As we've discussed before, this process is due to Coats, Rabs, and snares, and the vesicles will then be transported to an early endocytic compartment where the vesicles fuse. Contents of those vesicles are dumped out or remain membrane bound, and then they can either be transported down the endocytic pathway, or actually you have some molecules that get recycled, and we'll talk about this recycle path, back to the plasma membrane. Interesting features of this pathway is that the composition of each different compartment is different, and that means that the enzymes that are in the compartments, so the enzymes that are in each compartment are different. Some of them overlap, some of them are completely different, and the pH changes down this pathway. And that's going to be important because you're going to see that a lot of these enzymes that are in here are called acid hydrolases. All right, I expect you to know all these words. The question sort of comes up, well, why have different compartments? Why not just take things in from the outside to the inside and go directly to the lysosome? Well, why have different rooms in a house? There are different functions to each of the different compartments. As I said, vesicles come in from the plasma membrane. They are Oftentimes these are clathrin-coated vesicles that have clathrin, dynamin, adaptins, and they have rafts associated with them. The coat is lost, and then that vesicle can fuse with an early endosome. Here they're showing you ligands in red. So here are some ligands. Ligands are binding to receptors in black. The ligand-bound receptor is taken into a clathrin-coated pit, which buds off due to the action of dynamin, and the coat is lost, the blue coat is lost prior to fusion of this vesicle with the early endosome. The pH, right, so you should know that the pH, right, extracellular is somewhere between 7.0 and 7.4, so we're at neutral pH at the cell surface, but immediately upon entering the cell, you start to see a decrease in pH such that the pH goes from, the easiest way to think of this is 7 to 6 to 5 to 4. It's really 6.2, approximately 6.2 in an early endosome, 5.5 in a late endosome or multivesicular body, and in the 4 range when you get into the lysosome. How does pH change from one compartment to another? Well, pH is really all about the number of protons that are around. So the number of protons that are in a particular compartment determines its pH. And how can you imagine getting a proton into each compartment? Well, you're going to see that there's something called the vacuolar ATPase. All right, and that vacuolar ATPase is a proton pump. So that is a proton pump. And what does a pump mean? Pump means it's going against the gradient and that it requires energy. This is not a spontaneous reaction. All of those things you should know. And that we're using the energy of ATPase to pump protons into each of these different organelles. So multivesicular bodies, late endosomes, and lysosomes pump more protons in than an early endosome does. Why do we want to change the pH of these compartments? Well, it all has to do with the enzymes that are in those compartments. I'm going to show you this example, and we're going to talk a little bit more about this later, but here we're showing you the vacuolar ATPase. It can also be called a proton pump. This is pumping protons into the lumen of an endosome. All right, they're not saying whether this is an early endosome or a late endosome. The purple is showing you a receptor. Right, this is a receptor that was bound to the membrane, and this little red dot is the ligand bound to the receptor. So think about what had to happen at the cell surface. At the cell surface, the ligand bound to the receptor, causing a conformational change to that receptor. That conformational change in the receptor resulted in this particular receptor being placed into a clathrin-coated pit, which eventually budded off from the plasma membrane and was then sent along to the endocytic compartments. When the vesicle fused, right, so remember what happens if you have a vesicle fusing, right, it's going to then have its, whatever was in the lumen is now going to be in the lumen of the organelle. The receptor portion that bound the ligand is in the lumen of the organelle. So what might happen when this particular receptor is present in a place that has lower pH? Well, it's really interesting. You remember the whole concept, if the pH is below the pKa, what happens to proteins? You protonate the particular amino acids that can be protonated. So this can change the conformation of the receptor, thereby releasing the ligand at lower pH. So at higher concentrations of protons, the pH is lower. This changes the conformation of, right, changes the conformation of the receptor, and that allows the release of the ligand into the lumen of the early endosome. this process is played out over and over again in the course of different cells, different ligands, different receptors. But this is a mechanism whereby a ligand can be taken in from the outer portions from extracellular media. Let's say a hormone can bind to a hormone receptor. It can then be taken in to the cell, to an organelle, and the hormone gets released into the lumen of the endosome. What do you think the ultimate destination might be for that ligand? Well, if it's a hormone, you might think that that hormone is going to end up getting passed down through the early endosome, late endosome to a lysosome where it gets degraded. Okay, what happens to the receptor? Well, sometimes what happens to the receptor is the receptor actually will get placed into a recycling endosome and sent back out to the plasma membrane to do this function again. I mentioned the fact that there are different acid hydrolases, and this picture shows you lysosomes, but I want you to be crystal clear about this. Acid hydrolases are found in early endosomes, late endosomes, or multivesicular bodies, and lysosomes. Different enzymes are in different organelles. And acid hydrolases are enzymes that aren't functional until they reach a particular pH. So in this case, they're showing you pH 5, and I said you go from pH 7 to pH 6 approximately in early endosomes. So at pH 5, you are now really in a late endosome. And in this case, there are different enzymes. Nucleases. What do nucleases do? Nucleases chop up nucleic acids, so strings of nucleotides. Proteases chop up proteins. Glycosidase chops up, what is a glycosidase? Right? Carbohydrates. Lipase cuts up lipids. Phosphatases take off phosphate groups. Sulfatases take off sulfate groups, or sulfurs, and phospholipases chop up phospholipids. So all of these are dependent upon the vacuolar ATPase, and the vacuolar ATPase is dependent on ATP. So energy is utilized to pump protons in to activate these acid hydrolases. Well, why aren't they just active when they're made? Well, think about where they're made. They're made in the ER. And in the ER, they get transported to the ergic, right, and then to the Golgi, and then to the early endosome, etc. And do you want these acid hydrolases to be active along the exocytic pathway? No, you don't want them even active in an early endosome, at least these guys. And that means that we have a mechanism to turn on the activity of these particular enzymes using pH. All right. So I mentioned to you that we actually have these late endosomes are also called multivesicular bodies. Well, this is a really interesting process. Let me just make this smaller. And what happens is, all right, if I draw, okay, I'm going to draw, all right, I'm going to draw just, here's an early endosome. I'm going to make my late endosome really big just so that you can see. When a vesicle buds off and goes to the late endosome, maybe I should make this red. Okay, this vesicle can end up fusing on that late endosome membrane, and you're going to end up with something that looks like this. All right. So you end up with that membrane and everything that was on the inside of that vesicle. All right, so if this had a little protein that was in there, it would end up looking like this. Well, on the surface of these late endosomes, occasionally what happens is you get the budding in. Okay, let me move this up. Let me just make the whole thing smaller, and I'll bring it back up to size. But occasionally what happens is that you get the budding in of a vesicle, and so what that might look like is, oops, let me just take back that last thing I did. Oh, I can't. Let me see if I can erase. Nope, can't erase. Can I go back? Go back? Alright, sorry about that. So if I draw this again for you, what it might look like is that you have this invagination of membrane into, alright, so which side is which? That's what you need to think about here. So in this case, right, it would look like this. And if this continues to invaginate in and you were able to pinch off the membrane, what you end up with is something that looks like this. Oops, not this. Alright. You end up with an internal vesicle, and that is called an intraluminal vesicle. So this vesicle is called an intraluminal because it's in the lumen of the late endosome, or multivesicular body. Multivesicular because you're going to see you're going to end up with a whole bunch of these on the inside of the late endosome. And then now let's zoom back out. Alright, when I zoom back out, this EM image here, I love this EM image because it's showing you here are two vesicles that are en route to this multivesicular body, and I hope that you can see very clearly that you have some density inside. This is a multivesicular body, and these vesicles are fusing and they're being taken into the intraluminal space in vesicles. This whole multivesicular sorting pathway is regulated by a bunch of proteins. And I'm just going to show you some of these, and what is happening is the generation of this compartment on the bottom right is the invagination of the membrane, and how proteins or cargo, so here they're showing on the top left, you have some cargo that has to get into these intraluminal vesicles, and the thing that's going to regulate it is the addition of this ubiquitin molecule. And so proteins can be they can be monoubiquitinated. Ubiquitinated. That's a hard word for me to say. So an individual single ubiquitin molecule can be put on to a protein, and that directs the formation of this entire process. So I hope what you can see is there's lots and lots of proteins being added and regulated based on this one little ubiquitin, and that's what ends up leading this to be placed into the multivesicular body. The ubiquitin actually does get removed as the last step and gets reused in additional rounds of ubiquitinating other molecules. So what's the purpose of this? You might be saying, okay, what's the bottom line? The bottom line is you're making vesicles inside of a lumen of an organelle. Well, the purpose is that occasionally this process can be used to turn off molecules that are signaling from their intracellular domain. So this is a protein, right, that was taken in from the cell surface, and binding of a ligand to this can cause this. Think of this as a lightning bolt. Ooh, that's a bad lightning bolt. All right, so if this is signaling, well, to get it to stop signaling, the thing that we need to do is actually take it out of the cell completely and to put it inside the lumen of this organelle. We'll talk about a specific example of this in the next lecture. So that is, right, that is the endocytic pathway. Okay? If you were asked to really, so here's the plasma membrane, you have early endosomes, you have late endosomes, you have lysosomes, and the pH decreases as you go, right? So from the plasma membrane, you have a pH of approximately 7. Into here, you have 6.5, 5.5, 4.5 in a lysosome. And this is activating enzymes. And those enzymes are hydrolases. So what is a hydrolase? It is going to utilize water to help break bonds. So these acid hydrolases are nucleases. They're going to break phosphodiester bonds. Okay, so that's nucleases. Proteases, what do proteases have? They have peptide bonds. So proteases. All of the different enzymes are going to be breaking bonds, and those bonds that are breaking are the ones that we made in making those higher ordered carbon-containing complex molecules. So the purpose is to break down molecules so that in the lysosome we can then reutilize those components. We actually use those again, we recycle. So I like to consider the endocytic compartment a recycling compartment. Some molecules will get recycled from other places. Remember, there's the process of recycling molecules from early endosomes back to the plasma membrane, and that's actually called a recycling pathway. And there are two recycling pathways, one that happens from the early endosome, and this is very early in the process, so within 5 minutes. Okay, so that can happen as fast as 5 minutes after internalization. And then there actually is a process whereby molecules might be taken from a late endosome to what we call a later sorting compartment. And that is a slower process and could take up to an hour to multiple hours for recycling back to the plasma membrane. So recycling is going to be really important. That brings us to the other pathways of internalization, and not everything is taken up in a clathrin-coated pit. And one really interesting structure that has come about that we know of is called a caveolae. So this caveolae right here, this is a compartment that is found in cells, and it actually has a protein called caveolin. This is the picture on the bottom here is showing you the underneath of a plasma membrane, and underneath here you can see both a clathrin-coated pit, which I've drawn a blue arrow to, but there are also these membrane internalizations that are primarily composed of lipids and this protein caveolin. Some people say that these are flask-like structures, so they're saying up here that this looks a little bit like a flask. I don't know how flask-like that is. They're more like a round-bottom flask, but if you look at those, right, they're showing you that these membrane invaginations are occurring from the plasma membrane. And what's really interesting about these are made on lipid rafts. So part of lipid rafts is necessary to take in molecules through caveolae. How did we find out about this? It turns out that viruses are good at using this pathway to get into cells. And we've learned more about caveolae and caveolin from virus infection than we have from any other mechanism. I know that there are mechanisms known now, but they're not known to me. So just understand that these are an additional way, in addition to clathrin-coated pits, you can have caveolae and caveolin form into vesicles. So these will pinch off just the same, and then you have a caveolin containing vesicle, and those can then be transported elsewhere in the cell. Alright, another mechanism for internalization was pinocytosis. I told you, if you think of this as cell drinking, so I always think Pinot Noir is a nice wine that you would drink, so cell drinking. Cells need to drink. These are really incredibly small vesicles that are taken in constitutively, which means it's happening all day, every day. And so that means that you're taking in a lot of membrane, and in a particular cell called macrophages. Macrophages are part of your immune system, and they're constantly sampling the environment through this process of cell drinking. And they can ingest 100% of their membrane every half hour. That's a lot of membrane coming in, and that means you constantly have to be having membrane. So if you constantly are having membrane coming in, right, with vesicles, little tiny vesicles, you constantly have to have vesicles fusing with this plasma membrane. Otherwise, the cell would shrink away to nothing. And the last of these specialized forms of internalization is called phagocytosis, and phagocytosis has to do with the uptake of molecules, and phage is to eat. And phagocytic cells eat things, and there's two kinds. So macrophage are one type of phagocytic cell, macrophage. Another type of cell is called a neutrophil. And these cells, these phagocytic cells, are part of our immune system as well. They can take up foreign particles, and it really is the difference between us versus not us. Anything that is not you will be taken up as foreign. So even beads. So we use latex beads in the lab to feed to these macrophages. The macrophages see it as something that's not them. They will extend these pseudopods. So these pseudopods are membrane plus cytoskeletal filaments. There are actually filaments in here, which we're going to talk about shortly. And these filaments get made and pushed out, and they will enclose around, in this case, these are, in this EM picture that they're showing you, bacteria. And as they're closed, as the membrane closes, you end up with the bacteria in an organelle called the phagosome. So this phagosome is taken in from the outside. And you'll see dendritic cells, I put them here, they don't necessarily, they're not really good at phagocytosis. They do it, but they're the least good at it. Come in! And that leads us to the next video, which shows you the actual process of a cell chasing down a bacteria to phagocytose it. I've labeled this video The Chase. So what you're watching is, it's a neutrophil that is on a slide. These cells that are sitting out here, the cells that are here sitting statically are red blood cells. They're not very happy. This is not a normal shape for a red blood cell. But the neutrophil chased down this bacteria and has internalized it into a phagosome, and it will take quite some time for that neutrophil to complete eating it. Let's just watch that again. And let's start from the beginning. So this is the bacteria out here in front. This little guy that's being chased. I can't really circle it, it's so fast. There's some extra bacteria, but what's going on is you have the neutrophil chasing down that bacteria. It's following it based on a gradient of molecules that the bacteria is shedding as it's moving through the liquid here. And once the neutrophil catches up with it, it envelops it in a phagosome, and that spells the end of the bacteria. There's some specialized circumstances that we began to talk about with polarized cells, and in polarized cells you actually have a specific transport, an endocytic pathway that comes from the apical membrane, but you also have an endocytic pathway that comes from the basolateral membrane. And we believe that they meet somewhere around late endosomes. So there's only one set of late endosomes, there's only one set of lysosomes, but you can have apical early endosomes and you can have basolateral early endosomes. And this allows for some sort of sorting process to occur at this late endosome spot. And so just beware of this, that there is this transport, and it can be directed transport. Anything that's transported through the cell in a vesicular transport pattern is said to be going through transcytosis. That's trans, across. It's actually coming in one side, going from this side to the basolateral side, and ending up on the extracellular side of the basolateral side. And one of the specific examples is IgG. So the mom, when she's pregnant, can transfer antibodies. So IgG is antibodies, and it's a particular class of antibodies, to the baby, and those maternal IgG protect the baby from disease in the womb. This is done by transferring across the placental membrane, and that means on the mom's side it's the apical side, and they have to go from the mom's side to the basolateral side, which is the baby side. So we have specific transcytosis. Okay, what was the process going between cells? Okay, that was paracellular, P-A-R-A. All right, so you should be able to tell the difference between paracellular transcytosis and endocytosis. And so that leads us to the end of this lecture. We will talk more in detail with specific examples of receptor-mediated endocytosis, clathrin-mediated endocytosis. We won't talk any more about Caveolae-mediated internalization. It is interesting. If you're interested in that, search for viruses and Caveolae, and you'll get a lot of citations that give you information about that. We probably won't talk too much about phagocytosis or pinocytosis, but I'm going to give you some examples for this receptor-mediated internalization, specifically receptor-mediated endocytosis. Okay, that's the lecture for today. Hope you have a great day, and I'll talk to you soon.